This is the 2014 research update from the Chromosome 18 Clinical Research Center on Ring18. Here's a picture of Ring18. One copy of chromosome 18 is normal, and the other is formed a ring. And by definition, there's missing material. The caps that protect the ends of chromosomes must be missing off each end in order to form a ring. This slide shows a diagram of our molecular analysis for all the individuals in the study with Ring18. And across the top is a diagram of chromosome 18. And below that in the white box, you see all those gold bars. And every gold bar indicates the intact part of the chromosome for one person. And that person's study number is over to the left. So down the left-hand side, everybody's study numbers and their chromosome content off to the right. So the gold represents where they have 2 copies of chromosome 18, but the red shows where they have 3 copies of that part of the chromosome. So some individuals have major duplications of 18 as well as deletions of chromosome 18. The location of the centromere is the dark gold or brown vertical bar. And so you can see a couple people have deletions that include the entire P arm, but those same people have different deletions of the Q arm. So no 2 people are the same in this group, and in fact, they're not even close. Interestingly, you can see a couple people, for example, the very bottom person here, Ring18 13C, looks as if they do not have a deletion of the P arm because the gold bar goes all the way over to the end. And in fact, when we use FISH, another technique to determine whether or not there is a telomere at the end of the chromosome, we have a positive FISH result, meaning it detects the telomere, or it detects material very, very close to the telomere, out past the end of where the genes are. So the individual shown at the bottom actually doesn't have a deletion of any genes of 18P. There's also one person right in the middle of the diagram who also does not have a deletion of 18Q that involves any genes. They do have a deletion of 18Q, it just doesn't involve any genes. So you can see there's lots of variability here. And just so you can get an idea how this relates to 18P minus and 18Q minus, those people who have deletions of the entire P arm can be sure to listen to the report on 18P minus because there's a bunch of information there on people with the centromeric deletions of 18P. With regard to the Q arm, several regions are noted here. The distal 18Q minus region is shown here, so you can see everyone with a Ring18 chromosome and an 18Q deletion has a deletion that's somewhere in the range of somebody with 18Q minus. Also, the location of the TCF4 gene, which I'm sure you've heard about, is indicated there, and only one person with Ring18 has a deletion that includes the TCF4 gene. And then we had a recent paper about the reference group with 18Q minus, and we'll talk about that in the 18Q minus session, so you might want to listen to that. And so how that group relates to these people is shown here, so you can look at your child's deletion and see if they have a deletion of 18Q that's bigger or smaller than the reference group, so you can see where you are in regard to that group of people. And also, as we talked about in the introductory session, we talked about mosaicism, and that's particularly common in Ring18, and 53% do not have mosaicism. But in the almost half that do have mosaicism, we have all kinds of different conditions happening. 27% of the individuals have some cells in their body with one normal chromosome 18 and a ring chromosome 18, and some cells in their body with just a single chromosome 18. There's also some people who have Ring18 and a normal chromosome. That's some cells in their body. And the other cells have some are normal 18, but the second one is a derivative 18, meaning there's P minus or Q minus or some other chromosome 18 abnormality. Now, a very small percent have one cell line within their body that has a ring 18 and a normal chromosome, and the other cells are perfectly normal, normal 18, two normal 18s. And then there are another small percent who have three different cell lines within their body. The ring 18 cell line with a normal 18 in a ring, a cell line with completely normal cells, so two copies of normal 18, and then a third cell line that has only one chromosome 18 and no second copy at all. So there's lots of variability with regard to mosaicism within the ring 18 group. So this group, even more than all the others, has much individual variability. And we've not ever done an on-site clinical assessment of anyone with Ring 18, but we've carefully reviewed all the medical records, and this is what we've seen. And so you can read this list and see that the most common problem is growth hormone deficiency, which is hardly surprising because people with P deletions or Q deletions can be growth hormone deficient. So a group with both deletions is very likely to be growth hormone deficient. But here's the list of features that we've seen that are in the literature. In the adults with Ring 18, the most common behavior problem, and this is a small number of adults because it's a small number of people that have Ring 18, the most common problem is evading others to avoid social contact. So the good news is that there were no problems identified with a long list of behaviors. So for the most part, they really don't have behavioral issues. With regard to executive functioning or planning, which I think of as sort of the adult level planning of your life, your day, and your hour, deciding when you should start doing something, when you should stop doing something, how to organize things. That's executive functioning. So adults with Ring 18 had common problems in remembering the information to complete a task, keeping track of their problem-solving successes and failures, understanding what they're good at and what they weren't good at so they could do better next time. They had trouble moving from one situation to another and managing future-oriented tasks and modulating emotional responses. But most importantly, we need to focus on the problems they didn't have. They weren't acting out impulsively. They were appreciating their effect on other people, so they had really pretty good social skills. And they were beginning to do tasks independently and get themselves organized and their space organized. So they actually were acquiring some good adult sort of skills. With regard to social impairment, and this is in the children, so between 2.5 and 18 years of age, they had problems picking up social cues and interpreting those social cues appropriately. And obviously, social communication was an issue. And some of them had repetitive behaviors and obsessed on having the same routine or sameness in things. But few had problems with being motivated to engage in social behavior. So the younger children were much more outgoing. The natural history, and again, this is a very small group, so there's only 34 individuals in the study and only 5 over 18. And the information we had on their adult life is only from the 3 females. So one received a high school certificate, one a diploma, and one actually completed some college but didn't finish. All 3 live at home and aren't married and don't have children, and 2 of them have part-time jobs. And everyone always asks us about life expectancy, which is really hard to tell at this point. But what we can tell you is the current age of the participants in the study. And you can see they're a pretty young group, the oldest one being 45, but the vast majority being under about 12 years of age. 3 individuals have deceased with Ring 18 who were in the study. And you can see they died at fairly young ages, and their cause of death and the medical history about them is shown at the bottom, so you can have a little information about that. So Ring 18 is, as far as we know, isn't in itself a cause of early demise. And so our current and planned projects, pretty much everything we're doing relates to Ring 18. So we're looking at the effect of gene copy number on gene expression. We're interested in the consequences of genes that are already identified as dosage sensitive. And there's a study on 18P involved several of those genes. And so when we're interested in the effects of, let's say, a gene on 18P that we think is dosage sensitive, we don't just survey or inform the 18P minus families. We let any of the Ring 18 families know about it and see if they want to participate, if it's appropriate for them. So it really does involve Ring 18 families, even though it might sound like an 18P or Q specific project. It's really not. So everything relevant for P and Q is relevant to Ring 18. So thank you. And again, if you have any questions, please email Annis Hill, and she'll be happy to harass us until we answer the questions for you. So please let us know any concerns or questions. We always look forward to hearing from you. Thank you.